Hello everyone, I'm Rakshanda Sajeed and I'm representing my team from TCS India and will be sharing about our work combining learned protein embeddings and structural features for improved conformational epitope prediction. First of all, a brief introduction to the world of antibodies and antigens. Antigens are foreign proteins or their fragments that enter the host body via an infection and are capable of eliciting an immune response. An antibody is defined as an immunoglobulin capable of specific combination with the antigen that caused its production in a susceptible animal. The small site on an antigen to which a complementary antibody may specifically bind is an epitope or an antigenic determinant. These epitopes are usually 1 to 6 monosaccharides or 5 to 8 amino acid residues long and are present on the surface of the antigen. These are the proteins which play important roles in our immune system. The prediction of these epitopes is crucial for the development of vaccine subunits and therapeutics, and the development of computational methods for the prediction of these epitopes have thus been going on for quite some time. In this end, the language models have proved to be very promising. There are a few tools which are popularly used and have been stated here. These tools are machine learning and deep learning based. The protein language models are pacing ahead in this prediction task. In this end, BepiPred3 is the state-of-the-art method which works on protein embeddings from ESM2 for the prediction. However, we think that these PLMs might not be able to capture certain structural features which involve the shape and protuberance of the protein. Hence, our work involves utilizing embeddings from ESM2 and combining them with certain structural features of the proteins for the prediction of B-cell epitopes. Also, we explored different ways of combining the features and observe that deep concatenation of the features works better for our problem of interest. Now coming to the data that we have used. For our work, we have used B-cell receptor assay data from IEDB3D database for training, and we have taken the benchmark dataset used by Serendip CE tool as our independent test set. We filtered these protein sequences in these datasets based on their length and resolution of the protein structure. We've also made sure that these sequences in the training and independent test sets were no more than 25% similar to one another. This was done to remove the redundancy. For features, we utilize the ESM2 embeddings, and the structural features that we've considered are contact number, B factor, and protrusion index of the protein sequences. These three features represent the locality within the protein, its flexibility, and the shape of the protein. Thus, we have proposed two different kinds of neural network architecture-based models for this classification of B-cell epitopes, and in both of them we have used ESM2 as feature extractor, which gives a 1280 dimensional feature vector for each epitope or non-epitope point in our dataset. The first model here is shown where the 1280 features are simply concatenated with the structural features. This way of mixing different feature sets is commonly seen to work well in all settings. The final output representation passes through a 3-layer neural network where the weights of the neural network were learned for the classification of the residue into one of the two classes, conformational epitope or a non-epitope. In the second model, the ESM2 embeddings were transformed through the first 3 layers of the neural network. Then the transform feature vector was concatenated with different combinations of the structural features, which was passed through the final layer of the neural network. Coming to the results, we compared our model to the linear and conformational B-cell epitope prediction tools, namely BepiPred2, BepiPred3, and Discotope2. For each tool, we calculated the prediction values for the entire protein sequence on each amino acid. We compared the models using ROC and PR curves and scores. We also compared the models using balanced accuracy, and for this, we considered that if the average score of that verified region is greater than the standard threshold zero point 0.5, the prediction for that verified region will be 1, else it will be 0. Here we have shown the different metrics in the table comparing the performance of the models. We also trained our data using SAPROT, which is a more powerful and larger PLM trained originally on substantial protein sequence and structure data. We follow the same architectures for training and testing. We can see here an increase in the average precision score as compared to the models which had used ESM2. One can make an inference here that the three structural features are significant in characterizing the epitope residues. We've also tested an ensemble model of the MLP model, that is, the neural network trained only on the ESM2 features, and a random forest classifier model trained on the three structural features. We can see that the deep concatenation method still surpasses this ensemble way of using the features. Hence, our work shows that protein embeddings, when combined with structural features, lead to improved prediction accuracy as compared to protein embeddings alone, and that deep concatenation outperforms naive concatenation of embeddings and structural features. We can also see the importance of these three structural features. Our work and the results also highlight the biological significance of these features in classifying the epitope residues and their properties. Thank you.